Welcome back. We're thrilled you could join us for another episode of Technically Edinburgh. We are a brand new podcast created by technicians, about technicians, and for, well, everyone. Have you ever wondered what technicians do behind the scenes? What does that job title even mean? And what on earth is a technician commitment? We'll be chatting to staff across the university to find out why technicians are so essential to our work. We can't wait to get started, so grab a drink, get comfy, and let's get Technically Edinburgh.
Hello and welcome to Technically Edinburgh, a podcast for and about technicians at the University of Edinburgh. My name is Lee Murphy.
And I'm Charlie Corcoran. Our guest today is Edith Paxton, a core scientist from the Roslin Institute. She won the 2023 Technician Awards for contribution to research and innovation for her outstanding technical support and lab management and her contributions to research papers over the last 5 years. Hi Edith, welcome to Technically Edinburgh. Thank you so much for joining us. And firstly, huge congratulations on winning the 2023 Technician Award.
Thank you. Milking it here.
As you quite rightly should. So to kick us off, Edith, I was wondering how you would describe a core scientist.
It's just to support your PI and their postdocs and their starting students. So do any support. So basically keep them in line, make sure they know the health and safety, what to do with the waste, and just general things that you need to know about the institute, about where to get things, where to get your lab book and where to get your stationery and your lab coat and where to put it when you want to wash it, and glassware and all the sort of technical things, where you find a machine that you might need and that sort of thing.
So do you think of yourself as being like the institutional memory bank, that you've got all this knowledge of how Roslin works and you've been there for so long that you can help people?
Definitely. Everyone comes to me to say, do you know this? Or do you remember this person? Do you know whose initials this is? Whose writing this is on this tube? That sort of thing. Yeah. So yeah, that is one of the strengths of being older than everyone else.
I mean, you've said that. I guess I would say experienced rather than older.
I wonder if you could tell us a bit about your background.
So I did a BSc in biology at Napier University, although it wasn't a university then. And really, I did that because I wanted to be a vet when I was at school, but I didn't have the grades. So I think that happens to a lot of people. I used to drive past the Bush Road end when I was going to Edinburgh, because I lived in the Borders. And I used to think, I'd quite like to work there, because it would say, I can't remember what it said at the site. Now it says Veterinary Research Centres. And I thought, I'd quite like to work there. And then I had a job at what is now Charles River for a while. But then I met a friend on the street one day who worked at the vet school, and it was the Centre for Tropical Veterinary Medicine. And she said, I'm leaving my job to go and live in the Borders. You should apply for it. So I applied for her job and got it. And then, so that was Centre for Tropical Veterinary Medicine, where they do lots of things like trypanosomiasis and tyleria and lots of tropical diseases, obviously. So I had a few jobs there just because I got into the system and they wanted to keep me. So it was maternity cover and, people were going abroad for a while, so it was cover for them. So I was there for 17 years on various projects, all to do with tropical veterinary medicine. And then the funding came to an end. I worked on latterly I worked on a tick-borne disease, and my boss left and went to Kenya. So, and then the funding ran out. So I moved to, it was a vet pathology job, but it was actually in town. So working on a bacteria. So I did that for 3 years. And then I moved to the old Roslin to do immunology type job, but based with tyleria as well, which was a tropical disease. And all this time, so I worked part-time for 13 years. And then I came back to the vet school to a full-time job eventually, to a former PhD student who was now a lecturer. So yeah, and after, so that PI, he eventually left. I was with him for about 7 years, I think. And then Liam came from Glasgow. And I became his core scientist. So, and then that's been since 2013. So that's it, really.
That leads us to today.
Mm-hmm.
So I wonder if you could tell us a bit about your current role at the Roslin Institute.
Well, my current role is I'm a core scientist with Professor Liam Morrison, whose work revolves around trypanosomiasis. So we're involved in a few studies in collaboration with places in Tanzania and Glasgow and Liverpool and Nottingham, I think. So with field studies and things. So I support that work. We have 3 postdocs now, and I just support their work and generally do some lab management.
Just for anyone who's not sure, can you tell us what trypanosomiasis is, please?
Well, so it's just actually been changed to trypanosomosis.
Ah, okay, trypanosomosis. And it's a disease, right?
So it's the same as, you've probably heard of sleeping sickness in humans.
Yeah.
So it is the same disease, but whereas there's less than 10,000 cases of human trypanosomosis these days, there's still about 3 million cattle die every year of it, and it's a major problem in sub-Saharan Africa. It's a protozoan parasite that is in the blood, and it causes a disease in cattle called nagana. So it's transmitted by tsetse flies, causes a wasting disease in cows, and obviously it's more animal production, so they lose production, and it's usually poor farmers that only have 1 or 2 cows, and they don't give milk, and then the cows are got no meat in them by the time they don't breed if they're sick. So it is a major problem.
And what is the focus of your research work then on trypanosomes?
It mostly is drug resistance. So we do. So that was the field study was about. The most recent one was about drug resistance. So the project was initially farmers would be contacting the vets when they were going to dose their animals. So they would take a blood sample and then check if they actually had it, because they dose their animals anyway. They do it prophylactically. So it was just the vets were just making sure they actually had trypanosomes in their blood. They would record what drug they were given and checking they were diluting it properly, because quite often you can get cheap drugs at the market and they don't dilute them properly, and they're not given they don't give them enough, or they don't dilute them with clean water, and there's various things that can go wrong. Sometimes they get these drugs from China and the writing's in Chinese, so they don't know what it says, so they just sort of stick it in a jug with some water and then inject it into their cows without really knowing what they're giving it. So then they would follow up with a month and 2 months and 6 months after the dose. So that was how we got lots of blood from that. I think that's another paper. It's not actually finished yet, but that's another paper in the go. So it was to see, how the drug resistance, if it's because they're giving it to animals that don't actually have, the disease, and a lot of follow through. So a lot of samples that got sent here, and then that's a big part of the project I was involved in, was extracting the DNA from these blood samples and then doing PCRs. So there's 3 different species of trips. So you can do a PCR it's 4 different PCRs, but 3 PCRs it's for specific species, and then another PCR that's a kind of general one that you just sort of match up to the controls to see. So, and plus we did this in triplicate just because sometimes it just didn't work properly. So yeah, that was a lot of work. I think that's because I did all that sort of during sort of just after COVID and all that. So that was a major part of work. That's why I was nominated for this award, because I did thousands of them. And then also one of the postdocs that does a lot, we do like a gene's been identified that could be involved in the trypsin resistant. So he does a lot of genetic manipulation and things like that, which I'm involved in too. So he does all the the design of the experiments, and I just do things for him.
And is it always just one species that infects the cattle, or did you get multi-species infection?
No, there's the 3 species that infect them.
Can you be infected by all 3 at the same time, or?
Yeah, you can. Yeah, we culture them. We can culture them all, and so that helps with our in vitro stuff. We can culture the trips and, genetically manipulate them, transfect them with plasmids, and I'm not explaining that very well, but I'm sure you know what I mean. I'm sure people listen to our podcast will not.
Get you involved in the fieldwork at all?
Yeah, sometimes I get involved in the fieldwork. The main project that we've been involved in is a project in Tanzania. The Glasgow group are very involved in a group in Tanzania that with farmers, and they do, lots of blood sampling in farms and things like that. So we're involved in that. So I have been out to Tanzania a few times. I haven't been actually sampling, but I go out for I've been out to help students do various things. So.
That's very cool. It's exciting.
It is, yeah. I would like to go and be involved in taking the blood samples, but they don't really need me to do that.
Do you ever get scientists from Tanzania coming over to Roslin to work in your lab?
We haven't, no. Mostly, if they do, they go to Glasgow usually. Or Liverpool ones are involved in the tsetse flies. So we haven't so far.
One of the other things you were nominated for is your contributions to managing a lab space.
I know that it said somewhere that I managed a space for 15 groups, but I don't manage it. I kind of, I'm sort of the point of contact for people to complain about machines or whatever stuff goes wrong, things that go wrong. I'm the point of contact. I manage my own lab, and just to sort of generally keep on top of people being messy. You want to keep things tidy and clean, and just ask people to follow the rules a bit more often, put up notices and sort things out when they go wrong. I know who to contact and that sort of thing. So it's not really managing the whole lot, but we have a wing with about 15 groups in it. So I spend we keep a stock of consumable plastics and whatever. So there's a few of us take turns in doing that. So if people need any, plate, tissue culture stuff or pipettes or gloves or whatever, we're the ones to get it for them.
So roughly how much time do you split up between doing lab bench work and kind of the management side?
I just do it at the same time, really. If I've got experiments to do, there's always incubation times and whatever. I've always got time to order things and then pick them up at the stores and that sort of thing. I'm supposed to do 25% for the institute rather than just my PI, but I don't know if I actually do 25. I don't think many people do, but that's what we're supposed to do.
It's difficult to keep track of it, isn't it?
Yeah.
When you're doing just a little bit here and a little bit there, it's really hard to keep track of.
I don't hang about. Well, I do things. If I've got something to incubate, if I've got a 2-hour incubation, a 2-hour PCR or whatever, I can, there's plenty of time to do other things, really.
And is that how you're core funded then? Is that you get central funding from Roslin for all of that role, or is it partial of that role and some of it's on grant funding? I'm
not sure. I think I'm co-funded from Roslin. I don't know, because my boss sometimes says, that grant's, you'll be on that grant sort of thing. So maybe it's just 50%. I'm not quite sure how much is funded by it. As long as I get paid at the end of the month, I don't really care.
Absolutely. So I was just wondering if you have any strategies that you could pass on for maintaining organisation in that kind of like shared lab space.
My ideas were to make sure you have protocols for every experiment and then train new people and students in the basics of the lab. So if everyone's well trained, then nobody will get annoyed with them with doing wrong things all the time. Like the health and safety and where to put the waste and everything. And also to keep on top of your thing. We've got an awful lot of storage things stored in freezers. So always to keep on top of that is a good strategy. And get rid of stuff that regularly go through. We sometimes have a lab clean-up day, so you can go through all your boxes and throw things out and all your fridges and freezers. So, yeah, that's my advice to keep on top of people. These sort of things.
That's great advice for any lab environment. I think that was It's really good advice. So you were nominated for the award as well for your contributions to research papers. Are you happy to talk a little bit about that?
Yeah.
As the core scientist, the role that you play in the process of getting these papers out, where does that start and end?
It starts with me doing work in the lab, because I don't do anything to do with the writing at all. And sometimes, because I help quite a lot of other people as well, for instance, my boss used to share an office with someone who used to say, "Oh, would you mind extracting DNA from this blood sample," or whatever, and things like that. And then suddenly I'd be on a paper, and I'm like, I don't really know what I've done there. So because I was looking, I thought I better check how many I've been on. So I've been on 17 papers since 2016. And, some of them I have got no idea what I did. So unless it's just with I know what work I did for this blood from Tanzania. I mean, there's a few papers from that, and I know what I did there. I did the PCRs and I did the extractions, and another one we did specific work I remember to write a paper, and it was about a drug that we were using for the trypanosomosis, and Liam wondered if it might work with against tyleria as well, which is somebody else in our lab space works on tyleria. So I did a whole lot of work on that. So I knew that was for a paper right away, because I basically did all the lab work, and then somebody else wrote it. So, but normally, if it's my own lab group, I know that it's for a paper. But the ones that I do that I'm just helping people, sometimes I have no idea what I've done. And it's usually a few PCRs or a few DNA extractions or something.
And does that change? Is there a difference then in how you feel about being on papers that you work specifically, that you've done for the paper, and then just suddenly, oh, my name's on that paper?
Yeah, I don't feel so proud about them. I feel proud when my name's further up the list.
So would you say being recognised on so many papers has had any effect on your career?
It's not really done anything for my career. I don't think I've got any roles because of the papers I've been on, I don't think. Maybe if I had that many papers when I was trying to get a different job, because a job of somebody that didn't know of me, I think, might have been different. But
that's an interesting point. I mean, maybe not so much for technicians, but there's some pressure to publish and be recognised in papers. So how have you found your career progression, up to the last few years without so much recognition in papers to back up your applications?
Most of the jobs I've got, I've already been known to the person I'm getting the job with.
Right, okay, so it didn't matter so much if you didn't have papers to back up your application because the person hiring you already knew you and knew how good your work is. Have you found many challenges in your career?
So I worked part-time for 13 years, so it was this is a challenge, of course, when you have children, and then you have to try and get a part-time job, and you keep they'll say, oh, I can employ you, but I can only employ you at this grade 5 or whatever. So that was a challenge throughout my career. That's why I couldn't really call it a career, because funding kept ending, and then you'd move on to another job, and then we'd say, oh, well, we can't employ you in that. And everywhere I went, it was... So when I went from CTVM to pathology, I knew the guy that was employing me. I knew him. And then again, when I went to Roslin, I knew the woman who employed me. And then I came back to the vet school to a full-time job eventually, to a former PhD student who was now a lecturer. He employed me, but again, I can't employ you. I can only employ you at grade 5. But as soon as we get the funding for this, you can get upgraded. So these were my most challenges for not actually having a career. It was because I kept getting downgraded all the time.
It sounds like you've got a very wide scientific network now.
Yeah, well, it's very interconnected. If you're on the sort of tropical side of things in Edinburgh, it's just the same people sort of move around the country, I feel, until you decide to work on something else.
If you're good, then that's great, because then you can move from one team to the next.
Uh-huh,
because if you're bad, I'm assuming that then nobody would have employed you. They would have all...
Yes, I presume that. Yes, my boss is always saying I'm the glue that holds everyone together.
That's a nice thing to say.
That is a nice thing to say.
I make, when new people come, I take them up for lunch and take them for coffee, come and join us for coffee, and so people don't feel that they're on their own, and that sort of thing, and include people and cake days and whatever.
That's so nice. That's a really important role to play in your lab community.
Yeah, but it gets more difficult as you get older. They don't really want to go out with an old lady anymore. But as long as they come for coffee with me, so that's fine.
And it sounds like then that that kind of personal side is more important to you than, for example, the papers side.
Yeah, definitely. Yeah, because people are always saying, "Why don't you retire?" And I said, "Well, I quite like going to work. It's where else would you interact with young people unless you're a teacher, I guess." Yeah, so you interact with young people, and you wouldn't do that if I was just retired. What would I do with myself?
What kind of recognition have you had during your career, and how important was it being recognised for the award last year?
I don't have any recognition in the past, I don't think. Oh, I did get an award before, actually. In 2016, we got an award for just within Roslin. Again, it was a technician award. But then it's all down to lots of people look at these things and say, it all says, please nominate your colleagues or whatever, and everyone's going, oh yeah, I'll not bother. So it just comes down to who's willing to nominate someone. So my boss is very nice. He must have nominated me for that as well. But no, I've not really had any recognition apart from that, I guess.
Is that due to there isn't really those opportunities for recognition for technicians, or just in general, or is that, like you're saying, down to people being a bit apathetic and not nominating colleagues?
There's a few awards. They have them we have them in Roslin. We had since it's come more campus wide. We've had a campus wide one as well. But it is down to somebody to nominate you, I guess. And there's not that many technicians here. Most of the core scientists actually have PhDs here. So when it comes down to technicians, there's only a handful really, and obviously there's the people that do the services. They're technicians, but they're not involved in the science. They're doing glassware and the waste and that sort of thing.
Yeah, I guess it's up to us all to nominate our colleagues, isn't it? I guess you all need to put that effort in.
Yes, I think so. Yeah,
absolutely.
Mm-hmm.
And then part of Technician Week last year, you gave a great presentation at the event at Roslin. How did that go?
It was horrible.
Is that something you do often, presentations like that?
Well, luckily, it was only 3 minutes, and I was told I was very serious. I didn't mean to be serious, but I felt it was quite a serious thing to do. I'm not very good at public speaking. It's all a bit overwhelming.
But it went really well. I really enjoyed the talk. In Trypanosoma anyway, but I thought it was very interesting and kind of showed off your work really well.
Yeah, well, thank you. I still do. I'm not very. As soon as I have to talk about something, I forget it immediately, and then I end up reading it.
Oh, I have the same problem.
And then you end up reading it, you sound really monotonous. And
I think as technicians, we're probably not maybe as trained in presenting. Although I've had some interns this summer. And their presentation skills are amazing. I think what they are taught now in university is so much more. I think they do more presentations as part of their course, and they're much more natural at it, I think.
Yeah, it's quite. We have lab meetings, and I don't even present at that because I don't mind talking in front of my lab group, but because it's not really my work. I do work, but I'm doing it for somebody else, so I can't really talk about the background and stuff, which is another thing I should really learn about.
Yeah, it's that kind of lack of ownership. You don't feel like you own the results you've produced, that it's going to behalf of somebody else. And I think that's a real issue with technicians. And I do the same. I work in a core facility. I produce great sequencing data for people. I find it very difficult to talk about it because it's not, essentially my work.
And Edith, do you have any advice for other technicians who are maybe seeking to get a bit more recognition for their work?
Make sure you're interested in it and read more and contribute in lab meetings and things. Have an opinion about things and know the subject better. I feel and just helping people, help people do. Like I'm always asking people if I can help them do things and that sort of thing. And that gets you in the good books with your boss.
I guess you are now coming to the end of your career.
Yeah, I am.
When you look back, what you're kind of particularly proud of, or maybe frustrated about? How do you look back on your time as a...
When I was young at the CTM, I really enjoyed working there. It was, because there was a lot of us of the same, because I was younger, obviously, and there was a good turnover. There's always people, obviously PhD students come and go. They're 3 or 4 years or whatever, so always replaced by younger models all the time. So that's, yeah. But, these people that have been my friends all my life, really, so I've kept, in touch with them since I worked with them. So that's a good thing. Frustrating thing was, especially if you want to work part-time for a wee while, it's frustrating thing is getting... Fair enough, I got the job that I was in, I managed to wangle it so I could work part-time, but then every time you're trying to get a part-time job, it's always, it's just for this new woman, who's going to be rushing off to see her kids all the time, sort of thing. So I found that quite frustrating. And then that's why you couldn't really have a career if you had that big gap. I didn't really take it up again as anything seriously. I just, just had a job, basically. Wasn't really a career.
Do you feel that's getting better, or do you still see those issues with the younger staff members?
Well, it remains to be seen, actually. When I see some of the ones that are just having kids now and what happens to them when they come to the end of most of them are coming full-time. That take a year off and they come back full-time, so... or compress their hours or something. There's not many of them actually working part-time anymore, because you can't really have a career. I mean, I don't think, to be honest, technicians, I don't think, unless you're quite ambitious, you don't really have a career. You just have a job and try to get promoted as much as you can, sort of thing.
Well, thank you. I think that's a really honest answer. I think that's really interesting.
It's interesting from my point of view as well. I have young children, so I'm kind of battling that full-time children, what to do next, what's my career, what are my priorities kind of thing at the minute. So it's really interesting to hear.
I was just glad to get a job, really. And I don't know what else you could have done. You would have to retrain to do something else, and there's not really many opportunities to work part-time in this area. I think you probably could it all depends on your body, and you should be able to, because it's part of their equality and inclusion and everything, isn't it, to make it available to people. But in reality, it probably doesn't really work, and it does depend on your boss, obviously.
So as you're kind of approaching the end of your career, is there anything that you're still wanting to do, that you're still kind of excited about, that you want to maybe complete before you retire?
Not really, no. I'm quite happy to. I still would like to go to Tanzania another couple of times before I retire.
That's fair.
I'll see. And still, we also have links in Ilri in Nairobi in Kenya. And my boss, it was just before the pandemic, we were supposed to be going to Tanzania and Ilri, and we never got to go, so I've still not got my trip to Ilri, so I'm still looking forward to doing that sometime.
That would make a good retirement.
It would be a really good end.
If you could give your younger self a piece of advice, what would it have been?
I would say I would have read more, taken more interest in what I was doing, because I did kind of think about. My one boss said, I think you should do, he said an MPhil, because I didn't have an honours degree. He said I should do an MPhil, so I kind of started off with doing something to do with that, and then I got pregnant, and that was the end of that. And I didn't really fancy doing it after there. Do all your postgrad stuff before you have children. That's the idea. Or do have children. There's another one. There's another piece of advice.
It is a hard juggling act, isn't it?
Yeah, or just have them really early, and then I don't know what the best thing to do, because I don't feel that I've contributed that much until I started working full time again. When I worked part time, I was just had a job basically. So you can put more into it once you're full time. I feel. But then obviously not enough to make me want to read things.
Well, excellent. That's great advice for our listeners. Thank you very much, Edith, for agreeing to be on our podcast.
You're welcome. Thanks for having me.
Thank you so much for listening to this episode of Technically Edinburgh. I'd like to say a huge thank you to Lee Murphy, Imogen Hurd and Nick Kelly for their help in getting this podcast started. And of course to our incredible guests. Recognition and visibility are two of the key areas of the Technician Commitment and Initiative led by the UK Institute for Technical Skills and Strategy. It's so important to us that we make sure that technicians behind the research get the recognition and visibility that they deserve. And we hope this podcast is one small step towards that goal. Technically Edinburgh is hosted and edited and produced by members of the University of Edinburgh Technician Steering Committee. The music in this podcast is from Pixabay.com. Please follow us and subscribe wherever you get your podcasts. Check us out on X at UOE Technicians, and don't forget to tell your friends and colleagues. Have us on obnoxiously loud in your lab so everyone can hear. Oh, and if you have any ideas for topics, would like to come along and chat with us, or even think you might like to have a go at hosting, please get in touch at technician.network@ed.ac.uk. Thanks again to our amazing guest, our brilliant co-hosts, and you for listening. See you next time.